Oh. All right. Good afternoon, everybody. Today we are starting chapter 7, Fundamentals of Microbial Growth. So this week we're going to be covering part 1, pages 163 through 212. And so this is going to be the part of the chapter when we're talking about the things we need to do to promote microbial growth, okay? To get such lovely foods as sauerkraut and kimchi and pickles, okay? And then next week, we're going to do the rest of the chapter when we talk about controlling microbial growth, things we need to do to kill microbes in the environment. That we're spending a full week on because that's part of my professional background. And also I consider it to be something that is useful no matter what you're doing, either at home or in your profession. It's useful in all aspects of life. All right, so today we're going to go over the learning objectives. I'm going to open it up to questions. Then we're going to cover environmental conditions that influence microbial growth, specifically temperature, oxygen, pH, and osmotic pressure, if we get to that. And then we'll cover things to keep in mind for this week. All right, so this chapter will help you progress toward meeting the following learning objectives: being able to define the key role of evolution as it applies to microbiology, identify microbial structures and connect the structures to their functions, identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. All right, any questions? Okay, if you have questions later on, let me know. I'm happy to answer them. All right, so the first thing that we're going to do is we're going to talk about temperature and how that influences microbial growth. Okay, all right, so let's go to the whiteboard. So we're going to talk, that's not what I wanted. Stop it. There we go. So we're going to talk about temperature. So as a quick review, okay, let's go back to the infamous chapter 2 and talk about some chemistry a little bit. Okay, so let's say I've got part of a protein, and so this line is going to represent the primary structure, the line of amino acids. And let's say that here I've got a positive side chain. Here I've got a negative one. Over here I may have an OH that is sticking out. And here I've got another OH. Okay. So what's going to happen with that is that this positively charged side chain on the amino acid and this negatively charged side chain on another amino acid further down the chain are going to be attracted to each other. And so that's going to result in an ionic bond or ionic interaction. Okay. Here we've got 2 oxygens bonded to hydrogens. And so we're going to have hydrogen bonds that are going to form. Okay. So this is going to have a slight negative charge, a slight positive charge, slight positive charge, slight negative. And so we're going to get a hydrogen bond. Okay. So this is a part of what holds the protein together. Now remember, in this situation in the cell, we're in a water environment, okay? And all of these molecules are moving around, they're bouncing into each other and bumping into each other, okay? But these attractions should be enough to keep this part of the protein in its proper structure, what we call its native or functional state, okay? Now, heat or temperature, let's go with temperature. Okay, temperature is a measure of heat. Okay. And heat, for the purposes of what we're talking about right here, is the motion of the molecules. Okay. It's also an energy measurement. Okay. So as things are hotter, as there's more energy in the solution, things are going to be bouncing around more. Okay. And so as they bounce around more, okay, things are going to bounce into each other, and these are going to start breaking down. Okay. As water slams into this part, maybe this will move further over. And so this negative charge gets bent clear over here. Okay. This hydrogen bond gets broken. And what is the fancy dancy term that we have for when a protein is unfolded into its primary structure?
Denature.
Denature, yes. Okay. And sometimes when a protein denatures, it's able to pop back into its native state, into its functional shape. Okay. But oftentimes it doesn't. And so here is an example. Okay. Let's say this morning I got up and I felt like fried eggs. Okay. So I take and I crack this egg into my frying pan. Okay. What is it like when I first crack that egg? Is it a gas? Is it a liquid? Is it a gel? Is it a solid?
Gel.
Yeah, it's a gel. Okay. Kind of a jelly-like consistency. Okay. And then, I put some energy into the system. Okay. I light a fire underneath it. Okay. And I heat it up. It's going to go from a gel to a what?
A solid.
Very good. It's going to go to a solid. And that's because the proteins in the egg have been denatured. And so they formed a different shape. And when it cools off, does it go back to a gel?
Nope.
No, it stays a solid. Okay. And that's because the proteins in an egg are what we call temperature sensitive. Okay. So another fancy dancy term for this is temperature labile. But I'm not going to make you remember that one. Okay. For proteins that will pop back into their native or functional state, okay, we call them temperature stable. Okay, so I can heat them up and they will go back to their original form. Okay, unfortunately, prions that we talked about last week are temperature stable. You can cook them all you want, and they're still going to be in that problematic shape, okay, that causes functional proteins to bend into that banana shape. Okay, so why are we talking about that in this chapter? Okay, depending upon where a bacterium grows, okay, its proteins are in such a shape or conformation that they work best at certain temperatures, okay, especially when we talk about enzymes. Okay, structural proteins are also sensitive to temperature, but enzymes especially so. So I'm going to get my cartoon of an enzyme here, kind of our little Pac-Man shape. So enzymes have a particular temperature that they work best at. Okay? And that influences the temperature that the microbe will grow best at. Okay? So if I had a graph that showed temperature, okay, oops, I've got it going the wrong direction. This part would be cell numbers. and this would be temperature. As I'm growing microbes in the lab, I'm going to see them kind of, yeah, we're not happy. We're just going to stay asleep. And then when we hit a certain temperature, and it's different for each kind of microbe, then we're going to start seeing them wake up and dividing. And then they're going to plateau, and then they're going to drop off, okay, to nothing. So what happens is, if it's too cold for the enzymes especially, okay, to function appropriately, they just kind of stop working. It doesn't kill the microbe. Now, when you do that to humans, we die, but microbes are tougher. They just kind of go into hibernation, and they're just waiting for things to get warmer. So when it does get warmer, the enzymes start functioning. They're able to start growing and dividing, and we see an increase in numbers. Then we get to a point where it's like, okay, this is as good as it's going to get, and then it gets too hot, and their proteins denature, and they die. Okay, so we call this range through here the growth temperature. Okay. And right through here, okay, that's the sweet spot. We call that the optimum temperature. Okay. And different microbes have evolved to grow in different temperatures, depending upon which environment that they have adapted to. Okay. So let's do some examples. Okay. So let's say I'm in the Arctic, okay, and I am interested in microbes that live on glaciers, okay? And we're going to say that they're photosynthetic. Okay, so I'm putting some little green dots, okay, for photosynthetic bacteria that are growing on this glacier, okay? Now, the idea being that other photosynthetic bacteria that grew in warmer areas, maybe they got kicked up into the air and they happened to land on glaciers and on icebergs in Antarctica, okay? And those that couldn't live died. Those that were able to adapt and do well, okay, are now in this current environment, okay? So I'm going to go ahead and I'm going to get my cartoon of a boat set up here. And this is going to look a bit like Noah's Ark, except for we're going to put some, actually it's going to look like the Titanic. That's the problem. All right, so I take and I scrape these guys off, okay? So I want to take them home to the lab and I want to grow these puppies, okay? What temperature should I grow these microbes that have adapted to living on icebergs?
Probably around the same temperature as the iceberg.
Very good. Yes, we're going to want to grow them at the same temperature that the iceberg is at. Okay. What do you think would happen to these microbes if I tried to grow them at, say, human body temperature?
They might die.
Yeah, they would probably die. It would probably be too hot for them. And their enzymes that have adapted to doing very well at ice cold, literally temperatures, okay, are going to denature and it's going to kill the poor little bacteria. So the temperatures, okay, so we're going to go with Celsius, which I realize is a bit of a stretch, but we'll go ahead and we'll define these Celsius terms, okay? So ice freezes, actually, let me back up. Fresh water freezes at 0 degrees C. Okay, so this iceberg which calved off of a glacier, okay, is probably at about 0 degrees C. Salt water, especially in the Arctic, tends to be 4 degrees C, okay, because of all the salt in there. It takes a bigger drop in temperature before it freezes. Okay, so I would incubate them at about this range. Okay, so we would say that this was in the psychrophilic, we'll just go file, psychrophile range. Psychro for cold, loving. Okay. And so this range, this temperature range is an average. Okay. So of all of the microbes that we've isolated that live on glaciers and snow and in Arctic and Antarctic conditions, okay, when we look at their optimum growth ranges, we get this average, okay, this temperature range, okay. So that would be a psychrophile, okay. Now, let's say that I am also, okay, I've got an island off here in the distance, and it's a volcanic island, and we've got a little bit of smoke coming up here. It doesn't erupt, but you know, I've got a lot of steam coming up off of there, and penguins nest on these shores because they're generally ice-free, but we do get some good storms, okay? So I am interested in bacteria that live off of penguin poop, okay? Because, you know, that's what penguins do, right? So we have a range, okay? It's usually pretty warm here, but we do get some really severe storms through here during the winter, okay? So we would consider this seasonal. It gets pretty cold, but it's seasonal, okay? So for these troph, okay, and troph means eating, okay, so cold eaters. We have a range of about 0 to 30 degrees C. Now 30 is really warm. We're talking getting up into the 80s, okay, 80 degrees Fahrenheit. So these would we would call these psychrotrophs because they can grow when it's cold, but they do better when it's warmer. And they've got a broader range. So this is what we would consider seasonal, okay? Gets cold in the winter, but it gets pretty nice and warm during the summer, okay? So we've got that full range. Okay, now we humans, okay, I'm gonna go ahead and put a person here in in my ship, okay? Now let's say that we have an outbreak, okay, of say gastrointestinal distress, and because you know we're microbiologists and we're studying these microbes in Antarctica, you know, we want to find out what's causing a problem, okay, with our crew and our researchers, okay? So let's say we take some vomit, we take some feces, we're looking at the pathogen that's in there. What temperature should I incubate that at?
The psychotrophs temperature.
No, well, you would think so. Some human pathogens are psychotrophic. Yeah. But what's another good temperature if it's coming from the inside of a human?
Room temperature?
Yeah, some of them, you know, for this category that I'm getting at, some of them do grow very well at room temperature.
You talking about mesophiles?
Yeah, I am talking about mesophiles or middle loving. And of course, we humans have decided that the middle is the temperatures that we're comfortable with. So room temperature would definitely fit. Okay? But what if I wanted to look at, in other words, what I'm getting at is what is human body temperature?
97.5 degrees Fahrenheit.
97.5 degrees Fahrenheit. Okay. I'm going to believe you because I can't remember. But I do remember what it is in Celsius. See, here we've got 3 numbers to remember. Here we've only got 2. Okay, so human internal body temperature is 37 degrees. Now, if I were looking at oral or skin bacteria, because it's a little colder than it is, say, in the intestines, that's generally about 37. So if we're talking, 34. So if we're talking skin or in the mouth, they usually like it a little bit colder. Okay? But that would fit within the mesophilic or middle loving organisms. Okay. And in the book, they have a really broad range. Back in the day, we considered, well, let me tell you what we considered it to be. So about room temperature to normal body temperature, but the book has a different range. They say 10, which I'm sorry is really, really cold, and it overlaps with the psychotrophs, okay, up to 50, which is above a fever that would cause a person to go into convulsions. Yeah. So to me, this seems like a really broad range, but I'm not going to argue with the book. But back in the dark ages, this is what we considered mesophilic. Okay. So these are the temperatures that we consider to be in the middle range.
Which
Although
one should we remember?
remember the one in the book?
Okay.
Yeah. But when I write test questions, I'm never going to have them that low. I'm sorry, you put somebody at 10 degrees C without a polar suit on and they're going to die of hypothermia.
They aren't going to be happy.
No, because that, you know, it hasn't quite frozen yet, but we're in hypothermia range. Okay, so then let's say that I want to isolate, you know, bacteria that are living up here at the crater, okay, where it's really very, very hot, okay? And it's hot all the time because we're talking about a volcano. We call those thermophiles, okay, or heat lovers, okay? And the range for that one is 40 to 75 degrees C, okay? So in Celsius or C, water boils at 100, okay? So these guys like it hot. They generally are not human pathogens because we are just a little too cold for them, okay? So if they got inside of us, they would just go into dormancy. They would go into hibernation until things warm up. Okay? And then we have extreme thermophiles. Okay, and these are microbes that we have isolated from hot springs and from places where they are above the temperature of boiling. Okay, so let's say down here deep in the ocean underneath where I'm looking at the icebergs, I have thermal vents, and we call them smokers because they release minerals into the water and it looks like smoke. And the ones that have been studied most extensively, we call black smokers because they release a lot of sulfur containing minerals into the water. And sulfur, once it hits other minerals, tends to turn black. And so this is just because it's under pressure, it's at these autoclave temperatures, and we've been able to isolate microbes that are living here. And we have to keep them under pressure and at these higher temperatures. Okay, so these are extreme thermophiles. Okay, we've also isolated some from Yellowstone hot springs, all sorts of places you wouldn't think microbes would be living. But once again, it's where they've adapted to live. Okay, because, you know, all sorts of microbes are blowing around. Well, blowing isn't the right term. They're coming in the ocean's currents. Okay. And if, you know, say I've got one that's out here where it's not too hot and it doesn't get its proteins denatured, and so it survives and has kids, and those kids adapt and they get a little closer until finally I've got these microbes living at these extreme temperatures, okay, because they have adapted through random mutations, and the ones where the mutations benefit them, they're able to grow in an area where others are not there competing, okay? That's how we get microbes growing in every single environment we've looked at so far, okay? All right, any questions about these ranges? And why a microbe would be happy growing on a black smoker at 120 degrees Celsius, but would not be happy growing on an iceberg and vice versa. Okay, so come on. Oh, come on, let me do a new share. Why is my little thing not popping up? I suspect it's because I covered it, but there we go. We'll just do it that way. We'll just stop share because it won't let me give me my little thing. All right, so I'm going to ask you questions. I'm going to pull up a poll. Okay, so let's say that you have been cleaning your refrigerator, and you find in the back a container full of fuzz, mold that has been eating some forgotten leftovers. And suppose you have access to some potato dextrose agar, PDA, and a biosafety hood. And you inoculate the mold onto several PDA plates, and you incubate them at 0 degrees C, 24, 37, and 73. 75. And the grows well at 0 and 24. Which term best describes your mold? And I don't have an E, so don't pick E. And yes, there's more than one right answer. But you get to pick your favorite one. Okay, we're almost to 70%. Okay, we're at 76, 84. See if we can get to 100%. Okay, oh, there we got 92. Let's see if we can get 100%. All right, for the sake of time, yesterday I ran a little long. Okay, the favorite results are psychrophile and psychrotroph. Okay, those both would be good answers. Okay, but for the psychrophiles, they generally don't grow above 10 degrees Celsius. Okay, but we do have some bigger overlapping ranges in those temperatures. So if you pick psychrophile, you did pretty good, especially with being one of the first repetitions of the information. Psychotrophic is a better answer, okay, because that range is 0 to 30. Okay, so having it grow at 30 to 24 is right perfectly in that range. Okay, now if you picked mesophile, okay, that's also a pretty close to a correct answer. Okay, but mesophiles generally don't grow at 0 degrees C. According to the book, they grow at about 10. But so 24 degrees, mesophile would definitely grow at that. Okay. And thermophile, yeah, probably not. 0 degrees Celsius is a little cold for them. So B is the best answer for that one. Okay. Although psychrophile, maybe, mesophile, maybe. Okay. All right, here's another one. This one relates to lab. Okay, so we've been doing some experiments where we're looking at the endophytes, the fungi that live inside of sagebrush leaves. And based on the conditions that sagebrush around here experience and the temperature that we grew the mold in lab, the mold we isolated would be best described as which of these. And with this one, I'm sure we've got another couple of right answers. One of them's better than the others, but hey, we are almost to 50%. There, we just broke 50%. Ooh, we're at 70%. Okay, we're at 76. Let's see if we can get in the 80s. Oh, test all. Okay, for the sake of time, I'm going to go ahead and end the poll. Okay, the favorite answer was mesophile. Okay, good job. I would say definitely considering the conditions that sagebrush lives at, okay, and whatever the temperature outside is, that's the temperature that's in the plant. Sure, yes, because we range most of the time between 10 degrees C and, room temperature up to, more than what is comfortable, 100 degrees Fahrenheit sometimes. Okay, so an endophyte would have to survive those conditions. Okay? And then also, what temperature did we grow our little moldies at in the lab? Does anybody remember what degrees Celsius we grew them at? You're going, that was 4 weeks ago. That was a month. We put them at room 10. Oh, go ahead.
30 degrees?
Not quite 30, because that's a little toasty. We were more in the 25, 28 degree range. Yeah. So about 72 degrees Fahrenheit is what we keep the lab at. Yeah. So that would definitely be in the mesophile range. Okay. Now we have people picking psychotrophic. Okay. That is also a correct answer because the conditions that the sagebrush lives at fits within the psychotrophic range. Okay. In this case, mesophile and psychotroph overlap. Okay. So we would have to do further testing to see which temperatures it really prefers. Okay. And then if you pick thermophile, that's okay. We didn't grow them at quite that warm of a temperature, so I would say, yeah, probably not. But that's good for the beginning. All right, so I'm going to stop sharing and close that. Let us move on to oxygen. Well, I think I know what happened, why I couldn't get to. No, that's all. Okay. All right. So oxygen, we humans absolutely have to have oxygen. Okay. And in chapter 8, we're going to learn why. Okay. So oxygen, okay, when it exists in the atmosphere, two oxygens have a double covalent bond. So instead of sharing a single pair of electrons, we share two pair of electrons between the two atoms. Okay, so we breathe it in, our blood circulates it around our bodies, and then we exhale CO2. Okay. So to survive, we've got to have oxygen. Okay. And so back in the day, we didn't know why we had to have air. Okay? We just knew that if you cut somebody off from air, they would die fairly quickly. Okay? And so we use the term for microbes that have the same growth conditions, okay, that have to have oxygen. Okay, we talk about them being obligate aerobes. Okay, aero for air. Okay, we didn't know what part of air they needed, but now when we talk about obligate aerobes, those that have to have oxygen, and I'm going to abbreviate that as O2, to grow. Okay, so those are obligate aerobes. So those are microbes that have the same oxygen requirements that we humans have, okay, that our cells have, okay, because our cells have got to have oxygen. Now we have, because microbes have adapted to many environments, okay, they are able to grow in a variety of oxygen conditions, okay. So obligate aerobes have to have oxygen. They are obligated to have oxygen. Okay. Now, looking at the human body, there are areas of the body that have lower levels of oxygen than what we would see in the lungs. Okay. So over here in the stomach, okay, I've got some air because when I eat and when I drink, I swallow oxygen, okay? But it's not as high amount as there is in, say, in the lungs. The lungs are basically the same as what we find in the atmosphere. Okay. So there's less O2, okay? So microbes that we have isolated from the stomach, they're going, I can use a little oxygen, but a little oxygen goes a long way. You give me too much, you start to kill me. Okay? So we call those micro or small aerophiles. Okay, so they like a little bit of oxygen. Okay, and if you were in general micro, I would make you learn why. But in this class, you don't need to know. Okay, so then there are microbes that, if there is oxygen, they grow better. But they can grow slowly, okay, without. And in chapter 8, we're going to find out why. Okay, and we call those facultative anaerobes. Okay. All right. So there are other microbes that grow equally well with or without. Okay, and those are aerotolerant anaerobes. Okay, in other words, they don't use oxygen, but it doesn't kill them. Okay. So in the human body, especially in the intestines, okay, we have a combination of facultative anaerobes and aerotolerant anaerobes, okay? Because in the intestines, okay, there is effectively no oxygen. Okay. Any oxygen that your cells in the intestines are getting is coming from the blood. That's my story and I'm sticking to it. Okay. Now, there are also microbes that won't grow in oxygen or are killed by it. Okay. So have to grow in no oxygen. Okay. And those are obligate anaerobes. Okay. So in your intestines, okay, we have some obligate anaerobes. Okay. All right. Questions about these designations. The obligate anaerobes, the microaerophiles, facultative anaerobes, aerotolerant anaerobes, and whoops, obligate anaerobes. Okay, let me ask you questions. Okay, going back to our lab experiment, okay? Based on how we incubated the sagebrush endophyte fungi, the mold, in the lab, it would have which of the following oxygen requirements? Aerotolerant anaerobe, facultative anaerobe, microaerophile, obligate anaerobe, or obligate anaerobe?
For those of us who don't have lab, do you give us a vowel?
Oh, sure. Let me give you a vowel. Well, let me tell you what we did.
Okay, that would be great.
Yeah, because I keep forgetting that not everybody is in lab as well as lecture. So what we did is we took the sagebrush leaves and we put them on potato dextrose, and we just we put the lid on the petri dish, and we parafilmed them so that those spores couldn't get out, but oxygen can diffuse. Okay. And we just left them sitting in a cupboard in the dark.
Thank you.
Quite welcome.
Have they grown anything at all yet? It's been quite a few weeks.
It has been quite a few weeks, and we have gotten all sorts of fungi growing off of the early sagebrush. And we have had some very, very tough bacteria grow off of the Wyoming, but no mold. So there's a couple of things that might be going on. One, Wyoming is just toxic. Two, the mold, the fungi that live in the Wyoming, we're not providing the food on the PDA that it wants to grow on. There's a lot of microbes we can't grow in the lab because we don't provide the exact same conditions as what they are adapted to. So we're thinking it's one of those two things.
So they are growing, but they're growing slowly.
No, that we're not getting anything off of the Wyoming. It's not even growing
Nothing.
slowly.
All right.
Yeah. So
Yeah, because I
we're-
had the Wyoming D and nothing's happening.
Yeah, my section had the Wyoming D2 and nothing, a whole lot of nothing. Although we did send some of the bacteria that survived 4 minutes of bleach sterilization. We want to know what that is. That's scary stuff that's growing on the outside of that sagebrush.
Well, because I'm in your Thursday afternoon lab, and you only
that's
had
right.
one of the tables do that, right? The rest of us just did a 1 minute or a 3 minute or something.
And do you remember I showed you was that yellow growth that was
Yeah.
on the outside of the leaf?
Uh-huh.
Yeah, we sent that off for sequencing. Once we get the answers back, I'll let you know what that turned out to be.
Yeah, quite curious.
Yeah, because we didn't get it grow. We didn't get anything growing on the times that we dipped it for like 1 minute to 3 minutes. But on one of the 4 minutes, we had that one bacterium. So I suspect it's an endospore former. We would have had to leave it in there for 10 minutes to kill off any endospores. Yeah. So yeah, for the plates that you guys did a month ago, nothing, no mold,
Oh, wow,
Still.
still nothing.
still nothing. Yeah, I suspect we're just not feeding it whatever the mold would want. And so they're just kind of hanging out dormant, waiting for better times. And they're just not getting it. So they probably died by now. Yeah. Okay, we're at 100%. Okay, let me share the results. Okay, so with the favorite answer was obligate aerobe, which is a correct answer. Good job. Because we exposed it to air, and so we didn't cut it off from air, so we don't know if it's an obligate aerobe, but it could be. It could be because we gave it plenty of air. The second favorite answer is microaerophile. Okay, with that one, probably not. I mean, maybe there's microaerophilic conditions in there when we parafilmed it. So sure, I'll give you that one, sure. But generally to grow a microaerophile, we have to put it in like a closed container that we're feeding in just a little bit of oxygen. Okay, facultative anaerobe, yes, it could be facultative. What we would have to do to find out is to grow it with oxygen and without oxygen and see if they grow at different rates. Yeah. Or if they're obligate aerobic, they wouldn't grow without oxygen. Okay. So we would have to run an experiment to find out. But yeah, they could definitely be facultative anaerobic. And they could be aerotolerant anaerobic. Yeah. With that one, we would grow them with oxygen and without oxygen. And we would see if they grew at the same rate. In other words, they don't care. They don't care if there's oxygen there or not. Okay. All right. Good job. Good job. Okay, let's go on to the next question. Oh, C. diff. Clostridium difficile. Let's relaunch the poll here. Okay, so this used to be called Clostridium difficile. They've since changed the genus name to Clostridioides, but it still abbreviates to C. diff.
Was that fairly recently?
Yeah, that was within, I want to say, the last couple of years. Yeah. And some of the literature still shows it as Clostridium. Yeah, so what they'll do is they'll probably use both because, you know, people are used to seeing one versus the other. Yeah. But it is an obligate anaerobe that infects the large intestines, and it's associated with long-term diarrhea. Okay. Since it will only grow in an airless environment, how does it pass from patient to patient in an air-full clinical environment? So you've got 4 options. C. diff produces endospores, which are resistant to oxygen. C. diff will grow in aerobic environments, but much more slowly than an anaerobic. Fecal material will protect the bacterium from oxygen until it is swallowed and passed to the airless environment of the stomach. And it is very easy for C. diff to pass from patient to patient, especially if they're in a weakened state. So there's one that I consider the best answer. There's one that is like, yeah, I don't know, maybe. Okay, we're at 50%. And now we're at 61. And we're in 84. We're at 91. See if we can get to 100. There we go. Now we're at 100. Okay, so we had a good range of answers. The favorite answer is that C. diff will grow in aerobic environments, but more slowly than anaerobic. Okay, so far as I know, this is an incorrect statement. Okay, So far as I know, C. diff will only grow in oxygen-free environments. Okay, but that was good reasoning because we do know that it passes from person to person in a full air environment. So how does it do it? Okay, we had the next favorite answers. There was a tie between C and D. Okay, so D is a true statement. This is a true statement. And it's easy to pick that one because you're going, yeah, that's true. But it doesn't answer the question of how, if it'll only grow in airless environments, how it passes in an air-free environment. So you got to look for an answer that has oxygen in it. Okay. The next favorite one is C. You know, this could be true. I don't know. There, bacteria are microscopic. You've got microscopic amounts of feces around it that protect it. Okay. Well, I don't know. Maybe. Sure. But A, I know happens. Endospores are resistant to oxygen, and C. diff produces endospores, and endospores are really hard to kill. And that is the primary way that it passes from patient to patient to patient. Okay. The endospores are dormant. They're basically in hibernation. The oxygen doesn't bother them in that state. Okay. And so they are just waiting to go from one set of intestines to another set of intestines. That's kind of gross, huh? So C could be true. I don't know if it is or not, but sure, we'll say yes. But I know this one is definitely true. Okay. So this is why I emphasized endospores in chapter 3, because we're going to be seeing endospores a lot. Okay. All right, any questions?
No, but I'm also reading they can live in open air or in dirt for up to 2 years.
They can. And oh, go ahead.
Saying according to this website I was looking at, normal disinfectants aren't effective against them.
So wouldn't that mean B would be correct?
So they aren't growing, they're just in hibernation. Yeah, they're when we talk about growing, that means that they are metabolizing and they're dividing and making babies. Whereas endospores, they're asleep, they're waiting for better conditions.
So the key word in that sentence is grow.
It is grow. They will survive aerobic environments just fine. But they don't hatch out of their little endospores and become vegetative bacteria until they're in an oxygen-free environment.
It's one of those tricky questions.
Yes, well, I don't intend it to be tricky, but yes. Yeah, so yeah, and there have been in the lab, now this is in the lab, where they've had bottles of endospores of soil bacteria that do not cause disease normally. And what they used to do is they used to take little bits out and put them into airless environments, let them hatch out. And so far, we're at about 100 years with that same bottle. We've had some, yeah, isn't that scary? We've had some endospores that we've isolated from Egyptian tombs. Think King Tut, that were sealed for thousands of years. We put them in water, gave them a little bit of nutrients, they hatched out and were just fine after thousands of years. They're tough.
They're
They
like
are
sea monkeys.
Exactly, like sea monkeys. They can survive forever. We've sent them to space and exposed them to radiation. They don't care. So next week, when we talk about disinfection and sterilization, we will be talking about endospores again. Yeah, clinically, they make our lives very difficult. Yeah. But from the bacterium's point of view, endospores are an excellent adaptation to surviving lots of different conditions that would normally kill it. Okay. So yeah, C. diff, that's part of why C. diff is such a problem. Yes, Rosa. Oh, sorry, I saw that you had unmuted and I thought maybe you had a question. If you do have a question, let me know. Okay, let's go on to pH and osmotic pressure. And let's clear our board. Let me make sure that, okay, we've got half an hour. I can do this in half an hour. All right, going back to the infamous chapter 2. Let's get my part of a protein. Okay, and I'm just drawing part of it, you know, just imagine the rest of it going off this way and the rest of it going off this way. Okay, and I've got my positive side chain here. I've got my negative here. And I'm going to draw this a little bit differently so that the hydrogen bonding makes more sense. Okay. So in normal conditions, we've got ionic interactions here. We've got hydrogen bonds forming here, okay, that help hold the protein into its native or functional state, okay. Now we talked about temperature, okay. So what happens with pH? Okay, let's go ahead and get pH. Oh, that was cute. I want my whiteboard back. There we go. Okay. Where'd my controls go? Here, just a second, I gotta fix this. There we go. All right. So let's get pH and let's try not to freak it out again. Come on. Don't cover up my controls. All right. So pH is a measure of the concentration of free protons or hydrogen ions. Okay. So this is, let's say that this is in a close to a neutral pH. Okay. So the pH scale, excuse me, is an inverse scale. So the more hydrogens that I have in solution, it's acidic. And the smaller the number, okay? So from 0 to say 6.9 is considered acidic. My apologies, it's not my fault that it's an inverse scale. Okay, and then going from 7.1 down to 14, okay, you have less hydrogen ions or protons floating around, and we call that basic or alkaline. Okay, and right smack dab in the middle is 7, and that is considered neutral. Okay, this is the pH of distilled or deionized water. Okay, so in other words, just water. Okay, so let's say that we're going to take a human protein, because we're going to start with a macroscopic example that you're familiar with. Okay, and so the pH of most of our body, okay, is between about 7.1 to 7.3. Okay, so it's a little bit alkaline, but it's pretty close to neutral. Okay, now let's say, okay, that things become more acidic. Okay, I have more hydrogen ions floating around than we would normally have at a pH of 7.1 to 7.3. Okay, and what happens is these hydrogen ions start getting in the way. Okay, so I've got a hydrogen ion that comes over and says hi to this negative side chain. Okay, we get a hydrogen ion in here and it says hi, and it starts breaking these hydrogen bonds. Okay, and that will unravel my protein. Once again, what's my fancy dancy word for that?
Denature.
Denature. Okay, so if you get out of the preferred pH range of a protein, you can denature it. Okay, and you know, so let's get my person up here. Okay, So most of the human body, okay, is at this pH of 7.1 to 7.3, okay? But there are certain parts of us that are more acidic, okay? So let's take the stomach, for example. Okay, would we say that the stomach, inside the stomach, is acidic, neutral, or alkaline, basic?
Probably acidic.
It is acidic. Depending upon if you've just drunk a big glass of water or not, your stomach acid is between 2 to 3. Okay, so it's quite acidic. And the reason being is when you are digesting food, we put it in an acid bath to start denaturing and start breaking things down. Okay, it also kills an awful lot of microbes, okay, because their proteins are denatured. Okay. Now, before we go from the stomach to the intestines, okay, the intestines are not set up to handle this pH. So your liver produces bile salts, they accumulate in your gallbladder, okay? And so when we get to the intestines, okay, our pH is back to 7.1 to 7.3, okay? And your skin tends to be more acidic, okay, for the same reason. We want to keep the numbers of microbes on our skin down, okay? We tend to not be terribly alkaline, okay? Our bodies have not adapted to handling the problems that occur, okay, when there's not enough hydrogens. The exact opposite happens when you start sucking hydrogens out of solution, okay? They're not getting in the way, okay? But now we're sucking them off of these OH groups, okay? Because we've got so few hydrogens. And so instead of forming hydrogen bonds, we now have negative charges. This, instead of being hydrogen bonding to each other, are now repelling, okay? And that also denatures proteins, okay? All right. So we have microbes that live in these different ranges, okay? If they like to live in an acidic environment, so like if I were to isolate a microbe that lives in the stomach, okay, we would call them acidophiles. Or acid-loving microbes, okay? If they like to live in more of a neutral range, okay, and generally neutral, okay, is considered approximately this range, we call them neutrophiles. Okay. Now, so far we have not discovered many alkalophiles. Okay, but we have found some. Okay. And they tend to live in salt lakes, okay, because salty lakes tend to be more alkaline, okay, or what we call soda lakes. And so they tend to be very alkaline, very harsh conditions, not much growing there, but there are microbes that have adapted to living there, okay. All right, questions about pH, how it affects proteins, and what we call microbes that have adapted to live at these 3 ranges. All right, let's move on to osmosis. Okay, just for the fun of it, we're going to review what happens to our poor little bacterium at different salt concentrations. Okay, gonna go back to chapter 3. All right, so anybody remember what the salt concentration inside of a cell is?
0.8, right?
Close.
0.7.
Go up.
Oh, 0.9. 10.
Oh, no, I was wrong. Go down. If we round it, it's 0.01, but it's actually 0.09. And I wrote the right thing and I said the right thing. It's a physiological salt solution is 0.09. Okay. So when you said 0.8, I flipped a decimal, sorry about that. But I generally round it to 0.1. Okay. So we'll get rid of that. Okay, so let's say that this microbe is living in lake water. Okay. And in chapter 3, I would say that it had 0 salt, but really it just has really low levels of salt. Okay. Which direction is the water going to move? Into the cell, out of the cell, both directions equally?
Wouldn't it go into the cell because it wants to go to the party?
Exactly, it's going to go into the cell because it wants to go to the party. Okay. What keeps the cell from popping? Because we've got all this water flowing into it.
Bacteria wouldn't be cell walls.
Exactly, yes. The water pressure starts pushing on the cell wall holds it back and prevents the cell from popping. Okay. We, on the other hand, since we do not have cell walls, if you suspend red blood cells in a low salt solution, our red blood cells will pop. So when you get to A&P, you will learn how our body maintains a steady salt concentration. Okay? But we would call this a hypotonic solution. Okay? And most microbes do just fine in a hypotonic solution because they've got a cell wall. Okay? And just for the fun of it, what do we call a solution which is the same outside of the cell as inside of the cell? So we have, tell you what, I'll put the real non-rounded percent. Okay. And we've got a, okay. What do we call that solution? It's a wet solution.
Then an isotonic?
Very good. Isotonic. And where's the water going? This one's a little harder.
Doesn't it just kind of flow in and flow out?
It does. It flows in and out equally. Because water is just kind of bounce in, it's going to bounce out, but there's no, the party here is the same there, so you may as well mingle, right? Okay, now let's get to the reverse of our hypotonic. What would we call the reverse of a hypotonic solution?
Hyper.
Very good, hyper. It's supposed to be a C, not an S. Okay, so with my little bacterium, okay, we've got a 0.09% solution. And let's say we put them in a 2% solution. Okay, let's go ahead and give it its cell wall. Which direction is the water going to go? It's going to go in. Oh, yeah, it's going to go out. It's going to preferentially going to go out. And most bacteria have not adapted to survive this. Okay. So they turn into little, come on, they turn into little bacterial raisins. Their cell wall remains rigid, but their poor little cell membranes pull away from the cell wall and we get little bacterial raisins. We call this plasmolysis. Okay. So most microbes cannot survive hypertonic solutions. Okay. But there are some who have evolved, have adapted to salty conditions. Okay. So seawater is about a 2% solution. Okay. So I'm going to put seawater. Actually, I think I'm wrong. I think it's a 0.2. No, I think it's 2%. I think it's 2%. Don't quote me. Anyway, so, you know, if you're going to live in the ocean, you've got to be adapted to that. Okay. So interestingly enough, they have a number of ways of adapting to this, but their favorite way to do it is to actively pump water back in. So as fast as it goes out, they pump it back in, and they have to use energy to do it. Okay. But that's how they survive in a hypertonic solution. Isn't that cool? So microbes that survive in a high salt condition. Okay. So high salt. Okay. And they have to have high salt. Okay. Are obligate halophiles. Now, my standard joke has been, no, they're not addicted to the game Halo. That's Halo, not Halo. Now, getting my person back here. Okay. Are there any places on the human body where our little friends have to survive high salt? I go out,
your
I
body?
exercise, I sweat. Where does the sweat dry?
Your skin.
On your skin. Okay, the skin tends to be very salty. Okay, but it's not salty all the time. I get a fresh batch of salt, the microbes get more water, and they just, you know, so they survive high salt. They will grow in high salt, but they'll also grow in low salt conditions. Okay. And so we call those facultative halophiles. Okay, so your skin bacteria are facultative halophiles. Obligate halophiles we tend to find in seawater, okay, or in salt lakes. Okay. So these are the true terms that you need to know. Now, real quick, we've got like 2 minutes left. Okay. I've used salt, okay, as an example. The same thing happens whether we're talking salt or whether we're talking sugar. If we have a higher sugar solution outside of the cell, it's going to do the same thing. Okay. So when we're not specifying salt versus sugar, okay, we call this osmotolerance. Okay. So you can have osmotolerant microbes. Okay. So these are the microbes that you find growing on the top of that forgotten jar of jam that you opened 2 years ago and it got shoved to the back of the fridge. Okay. They are tolerant to the high concentration of sugar, so they can not only grow on it, but they can sit there and eat the jam. Okay. But next week, when we talk about preservation methods, back before refrigeration, what did we use? Salt and sugar. Besides, it's delicious. Okay, questions?
Can you go over how microbes survive in high salt again?
So generally speaking, at least with the ones I'm most familiar with, they have these transporters that will actively grab water and pump it into the cell. So as fast as the water is being sucked out of the cell through osmosis, they're pumping the water back in. Did that answer your question?
Yes.
Okay, other questions? Okay, we don't have time for me to bother you with questions. I shouldn't say bother. You probably think of it as, oh no, Teresa's gonna ask me a really hard question. Okay, so I've got some questions. So they're similar to the ones that are in the practice quiz for chapter 1 or chapter 7, part 1. Okay, so here's what's coming due. Okay, case study number 2. Unfortunately, I got most everybody's case study number 1 done. For those of you that I haven't, I've sent you an email. But for those of you that do have grades, take a look at my comments on case study 1 and use that to guide you as you do case study number 2, which is due this Friday. Okay. Exam 2 is also due on Saturday. Okay. And then to help you understand this chapter, I would strongly suggest going back and reviewing chapters 2 and 3 in the textbook. That'll really help with this chapter. Okay, any questions?